Today we're going to cover Unit 2, which is cell structure and function. Now to start us off, we have the processes of life. There's going to be 4 processes of life. The first one's growth. That means to increase in size. For example, the size of the organism or the size of the cell. B is reproduction. That's to increase the number. C, responsiveness, and that's the ability to react to the environmental stimuli, so anything that is going on around that cell needs to respond to or that organism needs to respond to. Metabolism, and that's going to be a controlled chemical reaction of organisms that provide energy and structure that is needed to grow, reproduce, and be responsive. So the term cell, or you could say cella, could mean a small room, and it's first going to be used by the scientist Robert Hooke in 1665 when he references an empty space or chamber. And he references this because it reminds him of prison cells and also of cells of the monastery rooms where the monks stayed. And so that's where he comes up with the name. So after observing these cells with their cell walls found in that dead cork that he was looking at, he actually decided that he was going to give it this name. So these, of course, they didn't have anything in them looking at these cork cells because they were from the bark of cork oaks and they were not living at this point. The cell theory was developed by 2 German biologists during the 1800s. We have Theodor Schwann and Matthias Schleiden. They both are going to state that all living things are composed of cells. When talking about cells, you can have 2 different types. You can have what we refer to as a eukaryotic cell or a prokaryotic cell. So we're going to cover the eukaryotic cells first. The eukaryotes are going to be fairly large for cells. They're going to be anywhere from 10 to 100 micrometers in diameter. They'll contain 1 or more true nuclei, each surrounded by a nuclear envelope, or you could say a nuclear membrane. They contain 2 or more linear chromosomes, so these chromosomes are going to be in straight lines. They have compartmentalized internal membrane-bound organelles that will do different jobs around the cell, and they're surrounded by a membrane with cholesterol and not involved in any type of synthesis or ATP in that cell membrane. They're going to be larger and more complex structures than a typical prokaryotic cell. Lots of times if you put a prokaryotic cell up beside them, you could fit hundreds of them on a eukaryotic cell. And these types of cells will include things such as algae, protozoa, fungi, animals, and plants. Now going to the prokaryotes, these are going to be these smaller cells. They're anywhere around, usually less than 1 micrometer in diameter. They don't contain a nucleus surrounded by a nuclear membrane or nuclear envelope. They contain 1 or more circular forms of DNA. And so these little circular loops are going to be covalently closed together. So it's not linear or in straight lines like it is with the eukaryotes. It's going to be circles for their DNA information. It can contain 1 or more loops of these covalently closed circular DNA strands as well. And sometimes it has a little loop. It just has a section of DNA, and those are often referred to as plasmids. They're not going to be compartmentalized by membrane-bound organelles, except for one. They do have ribosomes, which help put together different types of proteins that cell needs. They are going to be surrounded by a membrane lacking cholesterol, and its membranes also involved in ATP synthesis, wall synthesis, taxis, and other physiological activities. So smaller, less complex structures that are typically found in eukaryotic cells aren't going to be found here. And these will include things such as bacteria and the archaea. So now we're going into bacterial external structures. So here you will have a glycocalyx. That is going to be a gelatinous sticky substance that's surrounding that outside of the cell. It's made of polysaccharides. So poly means many, a saccharide is a sugar. So it's made of polysaccharides, proteins, and sometimes both, sometimes one or the other. And it is actually made by being secreted by that cell. So there's 2 different types or ways that these cells are going to be surrounded. They could have a capsule around them, which could be a thick, well-organized surrounding that forms around it. It's firmly attached to the individual cell, and it helps to protect the cell from the host's defenses, such as phagocytosis. It could also have a second type. It could have a slime layer. That's going to be loose, thin, it's kind of poorly organized. The cells are going to be dispersed within it. It prevents dehydration, so it helps that way, and it helps it to adhere to other objects. Now, several functions of the actual covering or surrounding around these cells. It could have food storage there, so primary function can allow them to collect and store food outside cell walls during favorable conditions and during long periods of poor nutrition availability. So it helps them to sustain and to live longer. Helps with attachment to smooth surfaces, so the capsule formed by, let's say, Streptococcus mutans is going to be a common inhabitant of the human mouth, so you find that in the mouth quite often, and it's largely responsible for the formation of dental plaque. That's going to be a soft, whitish, gooey material that's readily removed with dental floss. It can be helped and used for protection, so it protects it from predators, desiccation, which means drying out, and phagocytosis. That's where the immune cells will come in, surround that cell, bring it into the cell, and then dissolve it and destroy it. It also helps to increase pathogenicity, that's disease-causing ability. And so that capsule-forming bacteria are going to generally be ignored by the phagocytic white blood cells that are involved in defending us against different pathogens. Now, going into certain structures, we have the flagella. The flagella is going to be a long, thin protein filament that extends outward from the surface of the cell. It's going to be used for locomotion and movement. It's not going to be present in all bacteria. So with this structure, it's going to be composed, like we said, of a filament. It could have a hook and a basal body. The composed flagellum proteins and flagellins, these are going to be made up of linear strands that are going to wrap around one another, forming walls of the flagella. So the core of it is actually going to be hollow if you look at it there. It is not going to be surrounded by a cell membrane, and it's not supported by microtubules. It's going to actually attach to the cell membrane and the cell wall by a series of complex ring-like structures that are kind of similar to an axle attached to several wheels. It's going to have a basal body, which has anchor filaments, and those are going to hook to the cell wall by a rod and a series of either 2 or 4 rings of integral proteins. So looking at that, you can see those protein rings there in green that are going around. You see that rod going through the center, and all of that makes up that basal body that you see. Now, with the arrangement, you have 4 different types of arrangements. You can have what's called artrichous, which is going to be no flagella. You could have peritrichous, which is going to be a flagella that covers the surface of the cell. You can have polar, and that's going to be where the flagella are going to be only located at the ends. So you can have monotrichous, which is going to be a single flagella at the end. Amphitrichous, which is going to be 1 or more flagella at each pole. You can have lophotrichous, which is going to be multiple flagella at one pole. Or you can have what's called an endoflagella. With that endoflagella at the end of the spiral around the cell rather than within the medium. So it's going to form a axial filament following the cells to move via like a corkscrew type area. And so here's those different types that we just talked about. So like we said, we have that artrichous, which means no, the peritrichous, which is going to be cover the surface of the cell. So you see them all over. You can have the polar flagella, which could be a monotrichous, where you just have one coming out the side. It could be amphitrichous, where it can have one, let's say, on each side. Or you can have that lophotrichous. That's where you have that multiple flagella at one pole. Okay, so with the function, the motion of the bacterial flagella is going to be rotary. So they spin. It's going to be powered by ion flow. Usually it's going to be hydrogen ions. Sometimes it might be sodium ions that are going to cross the cell membrane. So rotation from that basal body is going to propel the bacterium through the environment. So it can be reversible. So it can actually go counterclockwise or clockwise. This is going to allow that bacteria to move in response to stimuli or taxis in the environment. So the movement is going to be in a series of, let's say, runs. So counterclockwise rotation in the same direction, and tumbles, which is clockwise rotation, in an abrupt direction. Next, we'll talk about the talk about the taxis there. So taxis is going to be movement in response to stimuli. So here we have stimuli can be light, so it could be what's called phototaxis. It could be chemical, which is called chemotaxis. It could be a magnetic field, which is magnetotaxis, or it could be gravity, which is geotaxis. So that movement is either going to be positive towards a favorable stimuli, or it could be negative, which means it's going to move away from an unfavorable stimulus. All right. Now going into fimbria. These are going to be sticky, short, hair-like appendages that are going to be found on the surface of some bacteria. They're going to be made of fibrin, which is going to be a protein, and they're often going to be quite numerous and cover the entire cell surface. They're going to be shorter than flagella, even though they kind of look like flagella. So let's say the flagella are going to be the long ones that you see there. You see the arrow pointing to it. The fimbriae are going to be the short hair-like structures. Now they are kind of longer than cilia, but shorter than the flagellum. Okay, so their functions are going to be attachment. So that's the primary function. That's going to allow the bacteria to bind to surfaces such as rocks and sticks, leaves, etc. Their environments, where they bind to one another, they can bind to each other using these, or sometimes in some cases of pathogenic forms, they allow them to attach to the host cell. Pathogenicity is another function of these structures here. For example, in gonorrhea, they're going to use this in order to be able to attach and become a pathogen to those cells. Cannot cause disease if they're unable to bind, though, to the surfaces of their host. So if they don't have these and are not able to bind, then they won't be able to cause that host cell to become infected. And they're also found in biofilms. So bacterial communities can attach to surfaces of objects, and often they are resistant to things such as disinfectants and antibiotics. Next, we have the pili. These are going to be special types of fimbriae. They're going to be longer and less numerous than fimbriae, but shorter than flagella. With bacteria, they typically are going to have 1 or 2 per cell. So they're straight, they're thread-like projections, and they are made of protein subunits called pilin. The function of these pili are going to be, number 1, for attachment. So it allows them to attach to bacteria, or to other bacteria, to other cells, to other surfaces. Another function is conjugation. And so that's going to enable one bacterial cell to adhere to another in order to exchange or transfer genetic material. So that's why pili are also known as conjugation or sex pili. So it allows them, even though when they divide through binary fission, bacteria are going to be single-celled organisms. They don't go through sexual reproduction. This is the closest thing that they have to it. So they'll use this pili in order to attach to another bacteria, and they exchange genetic information. That actually helps them to change up their genes some and to become more resistant to things that might normally would kill them. And the third function is pathogenicity. So that enables them to attach attach that bacteria to a host and become a pathogen. So next we're going to talk about the bacterial cell walls. So we have peptidoglycan. This is going to be a substance that is often found in the cell walls that surround bacterial cells. So bacterial cell walls are going to be rigid layers. Usually they're found outside of the cell membrane of most bacteria. They're composed of a unique material, like I said, called the peptidoglycan. And this is going to form a continuous network that goes around that cell. So it's basically a complex polysaccharide that's composed of two types of regularly alternating sugar molecules, and they're going to attach to other chains through amino acids. So this is going to form a single rigid mesh-like molecule that kind of looks like a chain link fence that goes around that cell. So the reason it does this, the functions, is to help maintain the bacteria's shape. So it has several different shapes depending on what type of bacteria it is. Sorry, that's going to show you that peptidoglycan structure, which kind of looks like a chain link. So going into these different shapes, you have your cocci, which is going to be a singular word as coccus. So cocci is going to be plural, and that's going to be the spherical ones. You have bacilli, or singular bacillus. That's going to be your rod-shaped cells. You have spirilla, or singular spirillum. That's the spiral-shaped cells. And then you have your vibrio, and that's going to be kind of like a comma-shaped cell. And so with these, you can have several different types of cell arrangements with these different cell shapes. So you can have the single that you see here. Sometimes it might be arranged in chains, sometimes in clusters, sometimes cuboidal packets. Sometimes you might actually see them come in and make like tetrads or cubes. You might be able to see them make grape clusters, all different types of shapes that they're able to make by attaching and arranging themselves. They can withstand what's known as Tugger pressure. Let's go back. And what that is that cell wall has that Tugger pressure so that cell does not lyse. So lysing is when a cell destroys itself basically or bursts. So this will keep that from happening. So that Tugger pressure is going to be the internal pressure that's created from the cell's contents. With bacteria, they're in a hypertonic solution sometimes, and so a more dilute solution is going to have less solute and more water than inside that bacterium, so it would cause water to kind of rush in and cause it to burst. So that would keep it from bursting. Another thing for this is going to be attachment. So it's going to assist in attachment to other cells, and it helps with the fourth thing, which is resistance. So it helps it to assist in the cell resisting antimicrobial drugs or anything that might come and kill that cell. So with bacteria, you're going to have two types of bacterial cell walls. You'll have what's known as a gram-positive bacterial cell wall. That type is going to be surrounded by a relatively thick layer of peptidoglycan. With that, it contains a unique polyalcohol called the teichoic acids covalently bound to the peptidoglycan. Probably this assists in binding positively charged molecules and surfaces into surfaces of cells. It's resistant to damage from physical factors such as heat, radiation, desiccation, which is drying out, and pressure. And when it's stained, it appears to be purple during gram staining procedures. Next, you have the gram-negative bacterial cell wall. This is surrounded by a thinner layer of peptidoglycan, and it's surrounded by an outer membrane. So that outer membrane is going to be based on a phospholipid bilayer and contains macromolecules like proteins, phospholipids, polysaccharides, specific ones that are going to be unique to these bacteria. They may be impediment to treatment of diseases, so it might be important to how they get in and how they're treated when they do actually cause you to become ill. They are more resistant to chemicals such as basic dyes, certain antibiotics, and lysosomes. So lysosomes are going to be the vacuole that's going to be found inside of the cell that contains enzymes that's able to break down and kill any type of microbe that gets into the cell. So it's kind of resistant to those. When they're gram stained, they do appear to be pink. So bacteria without cell walls will be rare. So they'll have certain characteristics that demonstrate relationships with other bacteria. They'll have cell membranes that are going to be strengthened as a result of cholesterols going through them. And then you'll also have a mycoplasm, which is going to be an example of these. So they'll have more phospholipids, they're going to have more cholesterol, things like that going through. Now moving on, we have bacterial cytoplasmic membranes. So with these, they are a living dynamic layer that's going to surround and limit the cell. Sometimes they're going to be referred to as an invisible surface layer because it's going to be too thin to be visible without a light microscope. So you have to have that type of microscope in order to see it. They're usually about 8 nanometers thick, and that's going to be considerably thinner than the skin on an apple or a color film that's going to be forming on the wall of a large soap bubble. So much, much thinner than that. It is said to form a selectively and differentially permeable barrier, and that's because it's going to allow only certain types of substances to pass through it, into or out of that cell, and because it's going to change over time. It's often referred to as a fluid mosaic model. Let's go back and look at this. So this is more of what we call a fluid mosaic model. This is developed by Singer and Nicholson in 1972. And so what it has is it's going to have a mosaic of lipids and proteins. It shows the phospholipids and proteins that freely move around, and it's going to behave more like a fluid than a solid. About 40% of this model is going to be membrane lipids. So they exist as the phospholipid bilayer. You see the little blue phosphate heads and the yellow lipid tails. So that's what it's talking about 40% of it. So phospholipids are going to have a hydrophilic head and a hydrophobic tail. So the blue is going to be the hydrophilic head, that's the phosphate, and then the tail, as you see over here, is going to be hydrophobic, and that's made up of the lipids. And so that's going to help in maintaining the structural integrity of the membrane actually itself. So membrane proteins are actually going to make up about 60% of the cell membrane. So they're going to be found floating in, inserted to, and sometimes penetrating through the bilayer. So you could have 2 different types. You can have what's known as integral proteins that are going to be embedded into the bilayer. Many will actually penetrate completely through. And you have 2 types of these that are embedded. You can have what's known as a carrier protein. It's going to bind to specific substances and transport them across the cell membrane. You could also have what's called a channel protein. These proteins are going to have a channel that runs straight through them, which is small, and it's water-soluble substances that are able to move across the cell membrane through these actual channels. The second type of protein is going to be referred to as a peripheral protein, and you can see that one, it's pointed towards the bottom. You have another one up there right above the word cytoplasm, but it's not pointing at it. It doesn't have like a indicator pointing, but that's one of those. So what they do is they attach loosely to the inner surface of the membrane. Some are going to be enzymes, some are going to be involved in electron transport chains or photosynthesis, and then others are going to be involved in changes in cell shape that are going to occur during cell division. Now with these, you might notice, different from a regular eukaryotic cell, these are going to lack sterols. And sterols, remember, are going to be your cholesterols that go through. Now, sometimes you have what's called membrane invagination. With this, that's going to be the folding in. So it's going to form structures that extend into the cytoplasm from that cell membrane. It's going to actually help to provide increased surface area for peripheral proteins or enzymes so they can catalyze chemical reactions. So you'll have 2 types of structures that are going to be formed from membrane invagination. You have these mesosomes. These are going to be membrane folds that extend into the cytoplasm that contain enzymes that are involved in ATP synthesis. Remember, that's your energy synthesis. It's also going to help to aid in chromosome separation during cell division. You'll also find them in thylakoids. These are going to be long, slender, membranous structures that are found primarily in bacteria that are going to use light as an energy source. So some general functions of the cell membrane. It's selectively permeable. That means most important function of the plasma membrane is that it can control what types of materials enter or exit that cell. And so you have 2 different processes of things coming in or going out. You have what's called the passive process that doesn't require the cell to expend any energy. Net movement of particles are always going to go down the gradient, so they always flow from high concentrations to low concentrations. And there's two types of gradients. So concentration or chemical gradient, that's the difference in the concentration of particles between one area and another. Or you could have what's called an electrical gradient. That's the difference in the net charge of ions existing between opposite sides of the membrane. So two examples of this passive process, you could have diffusion. That's going to be defined as the movement of particles from an area of high concentration to an area of low concentration. And you can have actually two types of diffusion. So you can have simple diffusion. That's the movement of chemicals from an area of high concentration to an area of low concentration. It doesn't require a membrane, but can occur across one. This can happen with gases such as oxygen and carbon dioxide when they move into and out of a cell through simple diffusion. You could also have what's known as facilitated diffusion. That's diffusion in which proteins act as channels or carriers that allow certain molecules to diffuse into or out of a cell along their electrochemical gradient. This does require a membrane because, remember, integral proteins are going to be necessary to help facilitate this transport process. So they have to be embedded into that cell membrane. So ions and small organic molecules like monosaccharides, amino acids, nucleotides, things like that, these are going to be the ones that are going to move into and out of the cell through this facilitated diffusion here. Remember, both of those are going to be your passive diffusion processes. Another type of passive diffusion is osmosis. Now, this is going to be defined as diffusion of water or a solvent from an area of low solute concentration, which is high in water, or to an area of higher concentration, which is low in water, through a selectively permeable membrane. So it basically goes from an area that's high to an area that's low. This is going to influence cell size and shape, causing a cell to shrink if water's leaving or to swell if water's entering. So excess water movement can actually cause cell damage, and sometimes it could even cause cell death. So you have what's called tonicity. That's going to be used in reference to fluid environments that surround cells. So equivalent to osmotic pressure, this is going to be pressure causing water to move by osmosis. So you're going to have three types of environments that we talk about tonicity. You could have an isotonic environment. That's where the solute particles are going to be equal and there's no movement of water. You can have hypotonic solutions. That's where solute particles are going to be less, so the movement of water is going to move inside. And you can have an hypertonic. That's where solute particles are high, so movement of water will be outside. So if you look here, the solutes are going to be what you see here as these little red dots. So the more little red dots you have, the more water is going to flow to them. So wherever the solutes are, water flows towards it. So if you look at A, it's got more of those solutes in there. And so B shows you how that water is flowing towards those solutes. So it gets more water flowing in. So if a cell has more solutes inside, water is going to flow inside that cell. If the outer environment has more solutes than inside the cell, then water is going to flow inside that, from inside that cell, out of the cell, causing it to kind of shrivel up. And so here's what you're seeing with the actual small cell up top, and you see with a what looks like a red blood cell almost, or some type of cell below. Actually, it's not a red blood cell, it has a cell wall. So this could be a plant or a fungal or bacterial cell below. So if there's going to be equal amounts of solutes inside and out, they keep their normal shape. If it's hypertonic solution, let's say there's more particles outside than inside, then that water flows out and will cause them to kind of shrivel up. If you see it where there's more solutes inside of these cells versus outside, water flows in, and it could even flow in so much it causes them to burst, and that is often referred to as lysing. So here we're going into the active process. With active diffusion, that's going to require a cell to expand energy in the form of ATP. Remember, ATP is your energy molecule. These particles are going to move against their concentration gradients. So we have two main examples of active process. We have active transport, and that's a process that allows cells to move particles through membranes against their concentration or against their electrical gradient. So that's going to involve integral proteins. And these protein complexes involved are going to be referred to as gated channels. It could be referred to as ports or as protein pumps. So you have three types of ports or pumps. You have what's called the uniport. That's going to move only one type of particle. This, for example, you see it moving. That could be, let's say, calcium moving out. You can have an antiport that's going to move two different types of particles in opposite directions at the same time. So that'd be like your sodium-potassium pump. And you can have what's called a symport. That's going to move two different types of particles in the same direction at the same time. So example of that would be like glucose with hydrogen or sodium ions. You can also have what's referred to as group translocation. That's going to be a mechanism allowing bacteria to move molecules through the membranes down their concentration gradients by changing them into different types of molecules during the transport process. Example of this would be glucose. So on top of allowing things into and out of the cell membrane, like we just saw, either through diffusion, passive, or active, it also helps to separate the cell from its environment. It helps with energy storage. It'll help harvest light energy in photosynthetic bacteria. It helps influencing taxis. So remember, taxis is going to be directed movement of cells within their environment in response to stimuli. Cell membranes are going to be equipped with various receptors. So stimuli is going to be perceived by these receptors, which help to trigger internal responses that result in the cell movement. Another thing that the cell membrane does is synthesize ATP and facilitate other metabolic processes. So you have ATP synthesis done through oxidative phosphorylation and phosphorylation. So that's where it takes in light to help do this. You're going to have enzymes that are going to be found in cell membranes that are involved with the following metabolic processes, such as synthesis of cell wall and constituents of proteins of the cell membrane. You'll have replication of DNA and formation of septa required for the cell divisions, and you'll have light production and some carbon fixation there. Another thing that you'll have with the cell membrane is going to be referred to as quorum sensing. So it involves receptors in the bacterial cell membranes that are going to be sensitive to signaling molecules released by other bacteria of the same type. So with these signaling molecules, you can have what's called autoinducers or pheromones. They're going to help to bind with membrane receptors and trigger the activation of genes that are influencing a variety of cellular functions. So we'll have the formation of more membrane receptors and like a biofilm formation that could happen. So it also help with light production, cell aggregation, a release of toxins, and the production of enzymes required for digestion of nutrients that cell will need. So going into bacterial cytoplasm. So there's some general cytoplasmic membrane functions that we'll see here. So with that cytoplasm, it's going to be gelatinous. It is made of a elastic material found inside of the cell. It's composed primarily of water, which is going to be about 90% of it, and then proteins. It's going to form the bulk of the cell. It's not going to be compartmentalized by membrane-bound organelles, and it's also going to lack structures that are going to be common in eukaryotic cells, such as the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, vacuoles, vesicles, and even mitochondria. So some components, it will have cytosol, and so liquid, that's going to be the liquid portion of the cytoplasm, mostly made up of water, plus it has some dissolved and suspended substances such as ions, carbohydrates, proteins, lipids, and wastes. It'll have a nucleoid. That's going to be found in the cytosol. It will be the control center of that prokaryotic cell, so that's kind of like the nucleus, but it's not going to be membrane bound. It's just floating around. It's not, like we said, going to be composed of its own separate space. So you can find it anywhere floating around inside of that cytoplasm, sometimes within that cell. So it's going to contain most of that cell's DNA and control the physiological processes going on inside that cell. Some other things you'll find inside the cytoplasm will be inclusions. These are deposits found within the cytosol of the bacteria. They'll store things like lipids, starch, and other chemicals. So some examples, you could have carboxysomes. They contain enzymes that involve fixing carbon dioxide or CO2 into organic compounds. You could have gas vacuoles or vesicles. These are going to be hollow cylinders that are surrounded by a thin layered protein, and they're going to be used to regulate things like buoyancy within the watery environment. You could have magnetostomes. These are going to be made up of crystalline materials, usually iron or magnetite, and they're going to be involved in the detection of magnetic fields or magnetotaxis. You could also have things such as endospores. These endospores are going to be unique. They're for dormant structures that are produced by some bacteria. They're going to be a defensive strategy against things like unfavorable conditions. So if, let's say, it's too hot or too dry, they can form an endospore, and it's going to help them be highly resistant to damage that's going to be caused by physical factors, let's say such as heat or pressure, radiation, desiccation, even toxic chemicals such as antimicrobial drugs. And so that's going to allow them to remain inactive but potentially viable for long periods of time. You could have some non-membranous organelles, two types of those that you will have: ribosomes, which are sites of protein synthesis in cells. They're composed of proteins called ribosomal RNA or rRNA. You also have a cytoskeleton. That's going to be the internal network of fibers that are going to play a role in forming a cell's basic shape. So you have spherical bacteria that appear to lack a cytoskeleton, but for most part, you will have a cytoskeleton sometimes. Now, coming down to plasmids. These are going to be small, extrachromosomal loops of DNA that carry genes not essential to cell function under most circumstances. The size of these can actually be variable, and these genes are going to carry a code for a variety of different proteins. And so these are sometimes what are exchanged through the pili between bacteria. So you see that plasmid being pointed at in the picture, that's just going to contain anywhere usually from 3 to 7 genes that are going to be on there, and that's to make some type of protein. Sometimes that might be the gene that actually makes that bacteria virulent or cause illness. Okay, so we just talked about bacteria. Now we're going into the archaea. So the archaean external structure, you can have what's called a glycocalyx. That's kind of similar to those on bacteria. They're composed of polysaccharides, polypeptides, or sometimes both. They do not appear to result in pathogenicity, and their function basically is adherence and biofilm formation. Next, you have your flagella. That function, in a manner similar to those in the bacteria, they consist of a basal body, a hook, and a filament. The difference between bacteria and archaeal flagella are going to be that they're smaller, not hollow. They're composed of different proteins. Frequently, they're going to have sugar molecules attached. They are powered by ATP, and they rotate together as a bundle when rotating counterclockwise. So that's the difference. Archaea has all of those. The fimbriae are going to be non-modal, rod-like sticky projections that are composed of protein, and they're going to help to anchor the cells to one another and to their environmental surfaces. And then the last, we have the hammy. That is going to be proteinaceous, which means it's made of proteins. The fimbriae-like structures are going to be there. They contain a helical filament with prickles and a terminus that's going to divide into 3 curved hooks. So you see that one up top. That's the hammy. The function is to securely attach cells to biological and inanimate surfaces. So it almost looks like a fisherman's hook that you see there. Now with the archaea, they're also going to have cell walls. So these cell walls are going to be composed of proteins and polysaccharides. They do lack the peptidoglycan. There'll be 2 different types of these archaean cell walls. You can still have a gram-positive and a gram-negative. Now with the gram-positive, Archaean cell wall, it's going to be thick and it's going to stain purple. With the gram-negative, it's going to have a layer of protein that covers the wall and it's going to stain pink. And it will have several different cytoplasmic membranes there. And so with these, they don't have a phospholipid membrane. They're going to be composed of lipids but lack phosphate groups. Some are going to have a single layer of lipid instead of a bilayer. And basically that function is to maintain the electrical and chemical gradients and control import and export of substances into and out of the cell. Now the Archaean cytoplasm is going to be a gel-like substance. And that's going to contain ribosomes, a cytoskeleton, and circular DNA chromosomes. They do not have membranous bound organelles. And the difference between bacterial and archaeal cytoplasm is that the archaeal ribosomes are going to contain different proteins that are going to kind of be similar to that of eukaryotic ribosomes. And they'll also have different metabolic enzymes that are going to be used to make RNA and genetic code. It's going to be similar to the eukaryotes. So with the external structure here, you have that glycocalyx that you're going to see on the outside. It's not as structurally organized as the prokaryotic glycocalyces. It's going to be absent in eukaryotic cells with cell walls, so you won't see this in things like plants, algae, fungi, or some protozoan. It's going to be present in eukaryotic cells that don't have cell walls, so you will see it in things like animals and some protozoan. The job is, or the function of it, is to strengthen the cell surface. So it helps to provide protection against dehydration, and it helps with cell-to-cell recognition and communication. So it lets, let's say, that cell be recognized by the immune system so it's not attacked. It's kind of like a fingerprint. Now with the cell walls, they're going to be a rigid layer that's going to be found outside of the cell membrane. These can be found in fungi, algae, plants, and some protozoans. They are composed of polysaccharides such as cellulose, it could be chitin, glucan, pectin, agar, but sometimes it can contain calcium carbonate, which is a glass. Their function is to help give that cell a characteristic shape. It helps with protection. It protects against things like changes in osmotic pressure. It could help with protection against predators. In plant cells, it'll be made up of cellulose. In fungal cell walls, it'll be made up of cellulose and chitin. And with algae in their cell walls, it could be made up of agar, alum, cellulose, calcium carbonate, or a combination of these different things. Next, you have your cytoplasmic membranes. These are going to be found in all eukaryotic cells. You have your fluid mosaic of phospholipids and proteins, which are going to be 50-50 ratio, 50% lipids and 50% proteins. You'll have a lipid bilayer that's going to be about 65% phospholipids, 25% cholesterol, and 10% other types of lipids. So they're going to contain things such as steroids. That helps to strengthen the membrane. It helps to solidify the membrane when temperatures rise, and it helps to maintain fluidity when temperatures fall. Also, it can contain membrane rafts. These are going to be compartmentalized regions that specialize in specific functions such as cell signaling, protein sorting, and cell movement. So with this, when it controls movement, it controls what goes into and out of a cell. So we can have endocytosis and exocytosis. With endocytosis, that's the active process requiring the expenditure of energy by the cell wall. It involves the invagination or inward folding of small sections of the cell membrane and the pinching off of that portion to form a vacuole or a vesicle inside of the cell. So you see that you'll have particles that bump up against the side. Let's say it could be a food particle or something the cell needs. As it bumps up against the side, it starts to bend in and surrounds it with that cell membrane. That is referred to as invagination. So it surrounds it with a membrane, brings it in, and then allows it to go to the part of the cell that it needs to go to. And it's surrounded by this nice phospholipid membrane that you see here. Now, it can be divided into two categories. If it is cell-eating or if it is a solid material, it's referred to as phagocytosis. That is used to move large amounts of particles into a cell. It's called pinocytosis or cell drinking if it's a liquid that it's bringing in. So it's going to utilize these particles if they're small or liquid molecules coming in. So that's referred to as cell drinking. The first one, phagocytosis, is cell eating. Both of those are an example of endocytosis, bringing things into the cell. Now, exocytosis is the opposite. Think of exo and exit. That's an active process requiring the expenditure of energy by the cell. So this process allows these cells to release various materials to the outside by joining those vacuoles or vesicles with the cell membrane from the outside. So let's say number 4 is moving up through the cell, comes to the edge. This membrane that's formed around these particles will then form with the outer cell membrane, and then it pushes these substances out to the outside world or outside of that cell. Now, eukaryotic cells can also have a flagellum. This is going to be found within cytoplasmic membranes, and it's composed of molecules of globular proteins called tubulin. They're going to be arranged in chains to form hollow microtubules and arranged around a central microtubule. So it's attached to a basal body. That's going to be surrounded by a cell membrane and supported by microtubules, and it doesn't have a hook and doesn't extend outside of that cell. So it may be single or it could be multiple. The function of flagella in eukaryotic cells is to provide locomotion or movement, to undulate rhythmically and push or pull a cell through a medium. It doesn't rotate and can exhibit taxis, so move from place to place. In a human, the only cell that's going to have a flagellum is going to be a sperm cell. Next, we have cilia. These are going to be short hair-like structures. They're numerous. They're often covering the entire cell surface. They move in a highly coordinated rhythmic fashion that create kind of a wave-like pattern that flows over the cell surface, or you can rotate around, let's say, food-getting structures. They can also be used to sweep substances in the local environment and to move materials along the surface of stationary cells. So you'll find these in places like the uterine tube. And so they're rhythmically moving and waving to move the egg from the ovary to the uterus, or you can actually find them in the airways that have mucus in them, and they're moving up to move that mucus up that has caught things that you've breathed in that could be pathogenic. So you move it up and away from the lungs. Hopefully, once it hits the pharynx, either you'll spit it out or you can swallow it and it'll go into the digestive juices of the stomach and be destroyed. So some other non-membranous organelles. You have your ribosomes, of course. That's going to be found within the cytosol as well as attached to membranes of the endoplasmic reticulum. So that's what gives the endoplasmic reticulum its name for the rough ER, because it looks rough and bumpy because of all those ribosomes that are attached. Now, they are a site of protein synthesis. So that's where all of your proteins are going to be brought to be made. You have your cytoskeleton. That's an extensive network structure that's going to be composed of both fibers and tubules. They're going to act as anchors to organelles, and they're going to function in cytoplasmic streaming and aid movement of organelles within that cytosol. So the function of the cytoskeleton is going to be to enable cell to contract, is going to be to help cytoplasmic membranes during endocytosis and amoeboid action. It helps produce the basic shape of a lot of cells, and it can help to anchor organelles. You'll have centrioles and centromeres. Animal cells and some fungal cells are going to contain two centrioles that are going to be lying at right angles of each other near the nucleus. And these are going to be found in centrosomes or regions of the cytoplasm. Their job is to play a role in mitosis and the formation of flagella or cilia for those cells. So here you see the different types of tubules that you can have. You could have the thicker ones, which are going to be called the microtubules. They're thick, hollow. You could have intermediate filaments. These are going to be durable rope-like systems. They help to give cell strength. And then you can have the smallest ones, which are going to be your microfilaments, which are going to be long, solid rod-like fibers that help a cell contract and help in that cell division. And so those are going to be your different types of filaments that are going to make up the cytoskeleton. Now with those centrioles, like we said, they're going to go to the different poles. They have what's referred to as the centrosome. That's going to contain both of the centriole pairs. During mitosis and meiosis, they're going to go to the poles and they start to emit that cytoskeleton coming out, which will then hook into the chromosomes and break them apart and pull them to either one side or the other side of the cell. Now here you see the nucleus. That's going to be a spherical ovoid, relatively large. It's centrally located. It's surrounded by a double layer of membrane. So it's not just a phospholipid bilayer, it's a double bilayer. So technically it's going to be 4 layers. It contains nuclear pores that are going to function to control the import and export of substances through the envelope. It contains a semi-liquid matrix called nucleoplasm. So it's like the cytoplasm of the nucleus. It contains chromatin, which are going to be thread-like strings of DNA that are associated with histone proteins. Histones are going to maintain the structural stability of the DNA and help to influence gene expression by binding with that DNA. And so during cell division, they're going to start to unfold and to actually make chromosomes. So they kind of tighten up to make those chromosomes, or when they're just floating around in the nucleus during regular day-to-day cell life, they're in that form of chromatin. So also inside the nucleus, you'll have a nucleolus or nucleoli. That could be one or more dark staining bodies, and it's going to be comprised of ribosomal RNA and protein. These are going to be the pacemakers of the cell because they influence protein synthesis, and they're going to be responsible for all of the protein synthesis that goes on in the cell because they make the ribosomes. So ribosomes are going to be made in the nucleolus. They control centers that are going to involve regulating the physiological process of making proteins also, and most of the cell's genetic material is going to be found inside the nucleus of the DNA. So the next organelle that you see here is the endoplasmic reticulum. It has a flattened, hollow, highly folded membranous organelle that is extended from the nuclear envelope into the cytoplasm and occasionally connecting to the cell membrane. This will include 2 membrane layers that are going to be separated by thin space. So it's going to take up most of the cytoplasm. It's a fairly large organelle. Now you can have 2 different types. You can have a rough or a smooth endoplasmic reticulum. Smooth is going to play a role in lipid synthesis, storage, and transport with some detoxification. Rough ER is going to be covered with ribosomes and they're going to be involved with protein synthesis. The Golgi body is going to be a series of flattened, hollow membranous sacs that are going to be surrounded by phospholipid bilayers. So they receive, process, and package large molecules in secretory vesicles that are released by exocytosis. They're also going to be the site of storage and primary site of polysaccharide synthesis. Lysosomes and peroxisomes. Lysosomes are going to be membranous organelles that contain a variety of digestive enzymes like hydrolases that are involved in hydrolysis. They are involved in catabolism of organic materials. So it's breaking things apart and putting things back together. They have a primary function within microorganisms that digest food and materials that are taken in through endocytosis, and they can help degrade worn-out organelles and break them apart and help to recycle any of that component that can be reused. The membrane vesicles that contain oxidase and catalase are going to be the peroxisomes. They're able to degrade poisonous metabolic waste such as free radicals and hydrogen peroxides. Now we have vacuoles and vesicles. So vacuoles and vesicles are going to be membranous organelles that store and carry substances. You can have a food vacuole through like pinocytotic vesicles. They're going to help to store nutrients that are ingested by protozoans. You can have a central vacuole. That's going to be large, centrally located region that's going to be filled with clear fluid. Lots of times these are found in plants and algae. These help to maintain turgor pressure within the cells and store toxins that might interfere with metabolism. You'll have a contractile vacuole. These organelles, don't have that on there. So these organelles are going to be found commonly in freshwater protozoa. They are circular structures that swell and shrink in the repeating pattern, function as a osmoregulator. They help to prevent cells from bursting due to osmosis or bringing in too much water. They also help to aid in fluid circulation within a cell, and they may have an excretory function. Next, you have your mitochondria. These are spherical to elongated organelles with two phospholipid bilayer membranes. So it has an outer membrane, which is going to be typical in eukaryotic cells, and then an inner membrane that lacks cholesterol and carries enzymes involved with ATP production. Now, mitochondria were thought to have been an organism that were brought into the cell that was once possibly a bacteria, and it just started functioning and working with the cell. The reason we think this is because it has that double membrane, one that matches the cell's membrane, but another that does not, and it has its own DNA. So the mitochondrial DNA will be different from the DNA that is found in the nucleus of the cell. So with these, they are found in most eukaryotes. They are self-replicating and often called the powerhouses of the cell. I know most of you, that's the one organelle that you can remember from biology. That's because they produce the ATP or the energy molecule. They contain ribosomes, they contain circular molecular DNA, and that's another reason that they think that it came, from a bacterial organization, because that DNA is circular and not linear like what's found in the nucleus of a eukaryotic cell. Our next one is our chloroplast. This is going to be your light harvesting organelle with two phospholipid bilayer membranes again. The inner membrane is going to be the thylakoids. They'll contain light trapping pigments that gather light energy to produce ATP and to form sugar from carbon dioxide. Now, it's found only in photosynthetic eukaryotes, so you'll only find these in algae and green plants. They are self-replicating, they do contain ribosomes, and again, these contain their own DNA as well. That DNA is going to be in a circular form, so the mitochondria and the chloroplast are thought to have been other organisms that were brought into cells and then just started to work with those cells. So that leads us to what we refer to as the endosymbiotic theory. That suggests to explain why mitochondria and the chloroplast have ribosomes, why they have circular DNA, and why they have two membranes. It states that the ancestors of these organelles were prokaryotic cells. So these prokaryotic cells were then internalized by other prokaryotes and then lost their ability to exist outside of their host, thus forming early eukaryotes. So they are widely but not universally accepted because it doesn't explain all the facts. So this was going to end us for our Unit 2 in micro. Hopefully my voice will quit giving out and we'll be fine for Unit 3. See you then.